Hello, my name is Shori Agrawal, and I'm a sophomore at Adlai Stevenson High School. Today, I would like to present to you my project, which is using supervised machine learning and integrative multiomics to predict pan-cancer drug response. First, I'd like to thank Ms. Palfi for always guiding me throughout the research process and always being there to answer all my questions, Mr. Clie for always answering my endless questions and giving valuable insights, and my parents for always motivating me throughout the process, and my mother for bravely battling cancer and inspiring me to take on this project. So currently, cancer is a leading cause of death worldwide. According to WHO, in 2020 alone, cancer caused 10 million deaths. And the problem is that current treatments include universal chemotherapy, radiation doses, and sometimes surgery. These are one size fits all treatments because they do not take into account disparities between individual tumors. This heterogeneity between the genetic information and biological processes of different tumors means that not all tumors respond similarly to the same drugs. And omics data can unveil and provide this genetic information for us to make individualized treatment plans for different patients. So now I'd like to talk about DNA. DNA, or deoxyribonucleic acid, is a carrier of genetic information that codes for various proteins. The main part of DNA that codes for these proteins are nucleotides, and nucleotides come in four types: A, T, C, and G. The way that DNA is turned into proteins is through transcription and translation, as seen in the image. In transcription, the DNA is first transcribed onto an mRNA molecule, and mRNA is transferred to the ribosome, where it's translated into proteins. This process of protein synthesis is regulated by various epigenetic factors. These factors include methylation and acetylation, where methylation will turn the gene off and block transcription from occurring, and acetylation will turn it on and cause it to occur. Next, I would like to talk about cancers and tumors. Tumors are an abnormal mass that undergo uncontrollable proliferation of cells, and cancers are a type of malignant tumor. These tumors occur when a mutation occurs anywhere along the protein synthesis process, whether it be on the actual genome, transcriptome, on epigenetic factors, or on the proteome. So, current treatments include chemotherapy, radiation doses, and sometimes surgery and immunotherapy. The problem with these are they are applicable across the board, and they end up eventually harming the patient because they do not respond the same to all patients. So now I'd like to talk about how omics data can solve this problem. Omics are various types of data collected to quantify and characterize different aspects of crucial biological molecules. In this project, I'll be using genomic, epigenomic, transcriptomic, and proteomic data. So these four types of data are essentially quantified, and their different aspects are quantified um, via the omics data that is collected. And in order for this data to be to uncover and unveil a larger view and more overarching complex patterns, we need to combine them together into multi-omics integrated data, and in turn that will allow us to look at a larger picture of of the process. So now I'd like to mention machine learning elastic net models. Machine learning is the ability of machines to learn complex patterns based on provided data to make predictions for new data. There are three types of machine learning, and today I'll be talking about supervised learning. And in supervised learning, the model is provided with features and labels, or inputs and outputs, and it tries to associate the inputs mathematically with the outputs. So when given a new set of inputs, it can apply the same pattern to accurately predict the output. So the problem with data, such as omics data, is it can be high dimensional, meaning it has a lot of features, since the human genome itself contains a lot of genes. And the problem with this high dimensional data is it can result in overfitting, where the model is less generalizable and cannot predict well on new given data. And rather than actually learning patterns, all it does is memorizes the training data. So, in order to reduce dimensions and reduce features, I intend to use the elastic net model, which can eliminate such data not required from the model. Another method to do this is via feature selection. Feature selection is a method to select the best features. These methods can either eliminate unnecessary features, meaning features that will harm the model or not be effective in the model, or reduce dimensions. Dimensionality reduction includes PCA. Feature elimination includes the methods mentioned on the slides. The previous research in this topic has been using only single omics to predict drug response. These studies lack data integration to combine omics types, and this led to low generalizability, where only sometimes a few cancers or a few drugs, a few subset of cancers or drugs were used. And combining omic types can lead to more complex drug resistance pathways and more overarching patterns to be learned and more biology to be learned. So my performance criteria was an accuracy, where the accuracy was an R squared of greater than 0.65, and it also focused on generalizability, which predicts effectively for at least two cancer types and for five different cancer drug classes, proving generalizability. Here's my procedure diagram. So essentially, my model first acquires data sets from the GDSC, genomics, proteomics, RNA, and methylation. The data is then wrangled, and then the data is then divided into training, validation, test set. Then the data is feature selected via variance threshold, select k-best, and from that, the input data is 660 samples for training, 82 samples for validation and test, 730 features or genes which were all RNA and methylation, since genomic and proteomic data were removed due to feature elimination, and 10 drug labels, which were chosen based on drugs that had the most data. This was passed into the model, which was a multitask elastic net model, and the output was AUC score prediction and accuracy and error. So now I'd like to talk about data wrangling. First, samples with nulls were removed, then the data was normalized due to the data being skewed. As clearly seen in both graphs, the data is right skewed because there's more data on the right side of the graph, and this essentially causes the model to be biased and have low generalizability. So in order for the model to perform better, the data was normalized. The data was also transposed to be compatible with the model. Then feature selection was applied on the model. Only simple feature elimination techniques were used, such as variance threshold, select k-best, to cut down low variance data or data that essentially had not a lot of effect on the model. And so before normalizing, this is what a correlation matrix looked like, where darker cells show that the input features are less related to the output labels, and the model explains less of the data, while for lighter cells, that means more of the data is explained by the model. So before normalizing, you can clearly see that the cells are much darker than after normalizing, meaning that after normalizing, the features were more related to the output variables and feature selection. So my models and results. So there was a multitask elastic net model. Hyperparameter optimization was performed manually. R squared was 0.47, MSE was 0.005. Model was successful in feature elimination as seen in the previous slide, and it also seemed to match common clinical information, as it tended to give most weight to genes that were clinically proven to have most weight during drug response. But also, this model was still high dimensional as seen with this graph over here, because there was still a lot of error and there was still a lot of the points not being described by the linear model. So in order to solve this problem, more sophisticated feature elimination techniques were used, such as forward SFS and dimensionality reduction such as PCA. Also, in order to better support or better fit the performance criteria, 8 different drugs were chosen from completely different pathways, targets, and clusters. The results from this were an R squared of 0.567, MSE of 0.0001, training R squared of 0.754. So essentially, this model had less overfitting and more generalizability because the training and test R squareds were much closer, and also better feature selection because the correlation between the output labels and the input features were much closer, as seen in this correlation matrix, as the cells are much lighter than with the previous model. Also, via coefficient analysis, you can see that a lot of the data is explained by the model. So my conclusion is that my model was able to discover some patterns, yet was not able to meet all the performance criteria, and this was due to several reasons based on the graphs before. The data was still somewhat skewed even after normalization, which caused the model to be biased. Overfitting still occurred, which caused the model to not perform well on test data. And the most important contributor of low accuracy was complex data. This data is extremely complex and has a lot of nonlinear tendencies along with a lot of noise, which cannot be often explained by simple models such as elastic net, which are linear. Furthermore, the data had a lot of noise. So my future research and Model 3 prototype, which I'm currently in the process of building, is a deep learning model which combines autoencoder and PCA to uncover more nonlinear tendencies and more linear tendencies to figure out the more overarching complex pathways and also get rid of more of the noise. Thank you for listening.